Hello students, in today's class we are going to learn about post-transcriptional modification. So the different types of post-transcriptional modifications that we will discuss include messenger RNA processing, including 3' polyadenylation, 5' capping, and splicing reaction. We'll also discuss about transfer RNA and ribosomal RNA processing. So the first slide is on polyadenylation. The polyadenylation reaction basically includes addition of stretch of adenine residue towards the 3' end of messenger RNA molecule. So messenger RNA molecule is normally synthesized beyond the polyadenylation site. Later on, it is cleaved by specific endonuclease by at a sequence which reads AAU-AAA in 5' to 3' direction. Once the new end is generated, it is further acted upon by a poly-A polymerase, which makes use of ATP and will synthesize a stretch of about 100 to 250 adenine residues towards the 3' end of messenger RNA molecule. So polyadenylation reaction basically helps to protect the messenger RNA from the 3' end from nuclease degradation, and also it is important during translational initiation process. The same points are written here for your reference. Next type of modification is capping. So capping is a modification that takes place at the 5' end of messenger RNA. So normally the bond formation that we have between the bases includes a 3' hydroxyl group and a 5' phosphate group, which are involved in the bond formation. But in case of a capping, we have involvement of a 5' phosphate of the 5' terminal base of the RNA molecule and a 5' phosphate of the external guanosine nucleotide. So as a result, the bond formation is actually 5' phosphate bond, triphosphate bond, which is formed. So as a result, since it is not like a normal bond, it also helps in protecting the messenger RNA by degradation from degradation by nucleases from the 5' end. Then the 5' cap is also important in transport of the messenger RNA molecule across the nuclear membrane by interaction with the nuclear pore complex containing the cap binding protein complex. Then once the messenger RNA has entered into the cytosol, then the cap also plays a role in translational initiation process. So looking at the steps of 5' capping. So here we can see the immature messenger RNA molecule containing a triphosphate at the 5' terminal nucleotide, and the base then undergoes first a loss of one phosphate group, leaving behind a diphosphate containing base at the 5' end of messenger RNA. Then we have entry of a GTP molecule, which will enter in the reverse orientation, so that we have bond formation between the phosphate residues itself, the 5' phosphate groups of both the nucleotides. So the GTP cleaves off two diphosphates during this reaction, and it results in formation of a triphosphate bond towards the 5' end of messenger RNA molecule. Further, the guanine residue is methylated in presence of enzyme guanine 7-methyl transferase, and here S-adenosine methionine acts as a methyl group donor. Sometimes you have additional methylation reactions where the ribose sugar of the additional one or two of the ribose sugars of the nucleotides are also further methylated. So the entire structure, including the 7 methyl guanosine residues and the additional methylated sugars, form the cap structure. Then we have the next slide is on post-transcriptional modifications in ribosomal RNA. So we have the pre-ribosomal RNA transcript that contains exon sequences for 18S, 5.8S, and 28S rRNAs. So these are first methylated and later on cleaved, and you have the mature 18S rRNA, 5.8S rRNA, and 28S rRNA molecules, while only the 5S rRNA molecule is synthesized as a separate transcript by RNA polymerase 3. Then during formation of mature ribosomes, which are made up of the larger 60S and the smaller 40S subunit, we have assembly of these different rRNA subunits. So the 60S is made up of 28S rRNA, 5.8S rRNA, and 5S rRNA, and 49 different ribonucleoproteins, while the 40S subunit is made up of 18S rRNA and 33 different ribonucleoproteins. Then coming to the transfer RNA processing. So a few bases towards the 5' end of the transfer RNA are cleaved off with the help of RNase P nuclease, while few bases at the 3' end are cleaved off by RNase D nuclease. Then further we have addition of a CCA nucleotides at the 3' end of the mature transfer RNA molecule. So the during formation of amino acyl transfer RNAs, we have addition of amino acids to the terminal adenine residue by the reaction which is catalyzed by the corresponding amino acyl transfer RNA synthetase enzyme. Then we also have towards the anticodon end, we have few more intron sequences which are also cleaved off during formation of mature anticodon arm of the mature transfer RNA molecule. Then coming to the splicing reaction, which takes place in the eukaryotic precursor messenger RNA molecules. So initially it contains both intron and exon sequences. Intron sequences are actually sequences that are present in between the exon sequences, which are the actual coding sequences. So during splicing reaction, these intron sequences are removed and the exons are joined together with each other, forming mature messenger RNA molecules. So 4 different types of splicing mechanisms are recognized: group 1 intron, group 2 introns, spliceosomal introns, and the 4th class of introns. So we basically have formation of a trans-esterification reaction during the splicing mechanism. So as you can see in the trans-esterification step, here either a 2' or a 5' hydroxyl group of the ribose sugar acts as a nucleophile, making a nucleophilic attack on the phosphorus at the splice junction, and in this process new phosphodiester bond formation takes place, as you can observe here. So coming to the group 1 splicing mechanism, it is observed in messenger RNA, ribosomal RNAs, and transfer RNAs of some nuclear, mitochondrial, and chloroplast genes. So in this mechanism represented here, we have the 5' exon and the 3' exon, and the intron sequence that is present in between. So in case of group 1 splicing mechanism, we have external guanosine molecule, the hydroxyl group, 3' hydroxyl group of which will begin the transesterification reaction by making the first nucleophilic attack at the 5' splice site. In the process, it itself forms a bond with the 5' intron, and a new nucleophile is generated at the 3' end of the 5' exon. So this newly formed nucleophile will now begin the second nucleophilic attack at the 3' splice site. In the process, it will itself form a bond with the exon, and the intron is displaced or removed in the process. Now, coming to the group 2 splicing mechanism. So it is found in case of DNA of algae, fungi, and plant organelles like mitochondria or chloroplast. So unlike group 1 splicing reaction, in case of group 2 splicing reaction, the base that makes a nucleophilic attack is basically an adenine residue itself, which is present within the intron sequence. Now, since the 3' hydroxyl group is involved in the formation of the RNA chain of the intron, so a 2' hydroxyl group is involved in beginning the nucleophilic reaction. So the hydroxyl group will make the first nucleophilic attack again at the 5' splice site. Forming in the process, it will form the bond with the intron itself, and the newly generated hydroxyl group will now make a second nucleophilic attack at the 3' splice site. Again, we have formation of exon bond in the process, and the intron is removed as a formation of a rope-like structure, which is also known as lariat. So in case of both group 1 and group 2 splicing mechanisms, there is no external input of ATP energy. Then coming to the third category, that is lysosomal introns. It is found in case of eukaryotic nuclear pre-messenger RNAs, and it involves a large protein complex which is known as lysosome, that is made up of several small nuclear ribonucleoproteins. And so 5 different snRNAs are involved in the process: U1, U2, U4, U5, and U6. So all these snRNAs, they basically form complex with the proteins to form the snRNPs. So 2 of the RNAs are involved in formation of a bond. So U1 snRNA has sequence that is complementary to the 5' splice site, while U2 snRNA or snRNP has a sequence complementary to a internal sequence that is present within the intron. So as you can see, because of the improper base pairing here, like all the bases are properly base pairing except the adenine. So as a result of which the adenine residue kind of protrudes outside, and as a result it is able to make the nucleophilic attack. So let us look at the mechanism. So we have first, during the splicing mechanism, we have entry of the U1 snRNPs and U2 snRNPs, which will bind at the 5' splice site and at the internal intron sequence respectively in presence of ATP molecules. So the 2' hydroxyl group of the adenine residue will make the nucleophilic attack at the 5' splice site. The rest of the snRNPs play a role by bringing the adenine residue in close proximity. So it will basically bring all the groups in close proximity with each other, And a very fast reaction will take place, wherein at the end of the process, again, the exon sequences are joined together and the introns along with all the 5 snRNPs are removed. So this is about the mechanism that involves formation of a spliceosome complex and also involves energy input in the form of ATP. Coming to the fourth class of introns, which is found in certain transfer RNA molecules. It requires activity of enzyme endonuclease and energy from ATP hydrolysis. So the endonuclease will cleave the phosphodiester bond at either ends of the introns, and the 2 exons are joined together, leading to formation of a new phosphodiester bond. So that's it about post-transcriptional modification. Thank you very much, and kindly like, share, and subscribe if you like the content. Thank you.